Hi, I'm BJ Demarine and I am currently studying at Mapua University. And I'm here to discuss about the optical characterization and elemental analysis of SEM with EDX. What is SEM with EDX? As it is stated here, SEM-EDX stands for Scanning Electron Microscopy for SEM with energy dispersive X-ray analysis for EDX. Essentially, SEM conducts with a high magnification microscope, which uses a focus scanned electron beam to produce images of the sample, both top and down, and with necessary sample preparation, can also produce images of the sample's cross-sectional area. In this figure, it shows the schematic diagram of the SEM-EDX process. It typically operates with a high vacuum that first generate primary electrons of low energy. Then secondary electrons emphasizes the topographic nature of the specimen. SEM may be equipped with an EDX analysis system that enables to perform composition analysis on the specimen. This analysis is very useful in identifying materials and contaminants as well as estimating the relative concentrations on the surface of the specimen. As you can see here, the specimen is bombarded with the electron beam. This is the electron beam inside the SEM, in which electrons collide with the specimen atoms of own electrons and knocking of them in the process. It can also backscatter to produce images with a high degree of atomic number contrast, such that ionized atoms can relax with the electron. Shell-to-shell transitions then leads to the either X-ray emission. This is the N-X-ray emission or an Auger electron ejection. These emitted X-rays are measured at a few micrometer of the sample by an EDX detector. Now here is an actual setup of an SEM-EDX machine inside the laboratory. And as you can see, this is the SEM-EDX machine connected to the computer or the software which will analyze the process that involves in this machine. And here are some results of a specimen test on the SEM-EDX with their corresponding analyzation. In the right figure is an SEM-EDX analysis of the nanofiber samples synthesized with pH equivalent to 6. On the left sample is a scanning electron microscopy and SED-EDX analysis of a slide. Lastly, here it shows an SEM-EDX result of a sample such as aluminum silicon glass and quartz in CR2-A. And so forth and so forth. Its main features, as you can see in that image, is that the magnification ranges from times 15 to times 200,000 with a resolution of 2 nanometer. It has an accelerating voltage from 1 to 230 kilo electron volts. With EDX, known or unknown materials can be analyzed. And lastly, qualitative and quantitative analysis for all elements are possible. Now let's go on the AFM. Now for AFM or atomic force microscopy, this works by scanning a probe over the sample surface, building up a map of the height or topography of the surface as it goes along. It differs from the other forms of microscopy as it no needs of focusing, illumination, and depth of field. AFM makes it simple by having height information which quickly measures the height, volume, and width of any feature in the sample. It uses a sharp probe to feel the sample surface, then builds up a map of the height sample surface, and lastly provides a single atomic level structure to provide a higher resolution. In this figure, it shows a schematic diagram of an AFM machine, which has 3 main components. The microscope stage, which has a moving AFM top, this, sample holder, this is the sample holder, and a force sensor. Second is a control electronics which has optical microscope and a vibration controller. And lastly is a computer which analyzes of all processes in that process. Thus, there are three different scanning methods or imaging modes for this microscopy. First is a contact microscopy mode, which is the fastest of all modes because of all repulsive forces that and sample may damage and that the top of the probe always touches the sample or in contact with it. Second is the non-contact mode, which is a signal-to-noise benefits associated with modulated signals as it has a weak attractive force, and these oscillating modes measures images with a small probe sample force only. Then lastly is a tapping mode, which has no capillary effect. Its amplitude signals are used in feedbacks, and these modes usually used for air imaging. There are only certain specifications of which AFM can only image, and such as maximum height on the order of 10 to 20 micrometers and a maximum scanning area of about 150 by 150 micrometers. The scanning speed of an AFM is considerably a limitation too, and that AFM machines are highly dependent on AFM codes. For some sample applications of atomic microscopy, here is some results of a high-resolution AFM imaging revealing the stoichiometry of an oligometric membrane protein complexes. It can also be used for samples like ceramic materials, human cells, or individual molecules of DNA and dispersion of metallic nanoparticles. Here shows an AFM images of BCTO ceramic sintered at a 1172 Kelvin for an 8-hour process, showing a 2D of grains and grain boundaries, a 3D image, and a histogram of a 3D particle roughness and a particle size distribution curve. And that's for all of it. Thank you for listening and thank you for watching.